The hippocampus is a structure in the human brain that plays a crucial role in the limbic system, being essential for functions such as memory formation and spatial navigation. Located in the medial temporal lobe, the hippocampus is an integral part of a complex network of brain regions that regulate emotions and behaviors. The limbic system is a set of structures that includes, in addition to the hippocampus, the amygdala, hypothalamus, cingulate cortex, among others. These structures work together to process and regulate emotional responses, being also responsible for motivation and behavior. Within this system, the hippocampus stands out for its role in memory and learning. Memory formation is one of the main roles of the hippocampus. It is involved in the conversion of short-term memories into long-term memories, a process known as consolidation. Without the hippocampus, the ability to form new memories would be severely impaired. This has been evidenced in various clinical case studies where damage to the hippocampus resulted in anterograde amnesia, the inability to form new memories after the damage. In addition to its role in memory, the hippocampus is also fundamental in spatial navigation. It helps create and store cognitive maps of the environment, allowing individuals to navigate and orient themselves in space. This function was initially demonstrated in studies with rodents, where it was observed that specific cells in the hippocampus, known as place cells, activate in response to specific locations in the environment. Neuroscientific studies reveal that the hippocampus possesses significant synaptic plasticity, meaning that its neural connections can strengthen or weaken in response to activity. This plasticity is fundamental to learning and memory processes. In the long term, changes in synaptic efficacy, such as long-term potentiation, LTP, and long-term depression, LTD, are mechanisms through which the hippocampus processes and stores information. The hippocampus does not work in isolation. It interacts with other brain areas, especially within the limbic system. Communication between the hippocampus and the amygdala, for example, is crucial for the formation of emotional memories. The amygdala processes the emotional aspects of experiences, and then this information is integrated into the hippocampus to form memories that have a strong emotional component. The hippocampus also communicates with the prefrontal cortex, a brain area involved in planning and decision-making. This interaction is vital for working memory and the ability to use past information to influence future decisions. Moreover, communication with the hypothalamus helps regulate physiological responses to emotions, such as the release of stress hormones. Disorders in hippocampal functioning can lead to various neurological and psychiatric conditions. For instance, in Alzheimer's disease, one of the first areas affected is the hippocampus, explaining the memory problems characteristic of this condition. Depression has also been associated with changes in the hippocampus, where reductions in the volume of this structure are observed in depressed individuals. Neurogenesis, the process of forming new neurons, occurs in the hippocampus throughout life. This is particularly interesting because it suggests that the hippocampus maintains the ability to adapt and reorganize, even in adulthood. Neurogenesis in the hippocampus has been associated with learning processes and stress resilience, indicating a role in maintaining mental health. Studies indicate that environmental factors can influence neurogenesis in the hippocampus. For example, physical activities, enriched environments, and cognitive stimuli are known to promote neurogenesis, while chronic stress and depression tend to suppress it. These findings highlight the importance of a healthy lifestyle for maintaining hippocampal functions. Additionally, the hippocampus plays a role in regulating the hypothalamic pituitary adrenal (HPA) axis, which controls the stress response. Activation of this axis leads to the release of cortisol, a stress hormone. The hippocampus has cortisol receptors and, under normal conditions, helps regulate the release of this hormone, promoting recovery after stressful situations. However, in conditions of chronic stress, this regulation can be impaired, leading to elevated cortisol levels and damage to the hippocampus itself. Research on the hippocampus has advanced significantly with the use of modern neuroimaging techniques, such as functional magnetic resonance imaging, fMRI, and positron emission tomography, PET. These techniques allow visualization of hippocampal activity in real time and study its connections with other brain regions. These advances have provided a more detailed understanding of how the hippocampus contributes to memory, spatial navigation, and emotional regulation. Understanding the hippocampus is also crucial for developing therapeutic interventions. For example, brain stimulation strategies such as transcranial magnetic stimulation, TMS, and transcranial direct current stimulation, TDCS, are being explored to improve hippocampal function in conditions like depression and Alzheimer's disease. These approaches aim to modulate neuronal activity in the hippocampus, promoting synaptic plasticity and neurogenesis. The hippocampus has a complex internal architecture with several subregions, including the dentate gyrus, Ammon's horn, CA1, CA2, CA3, and subiculum. Each of these subregions has specific functions and distinct connections. The dentate gyrus, for example, is one of the main regions where neurogenesis occurs, while CA1 is crucial for transmitting information out of the hippocampus. Neurogenesis in the dentate gyrus is a highly regulated process involving the proliferation, differentiation, and integration of new neurons into the existing network. These new neurons are considered important for cognitive flexibility, allowing the brain to adapt to new information and experiences. Research on how neurogenesis can be modulated has significant implications for treating cognitive and emotional disorders. The hippocampus is also influenced by several neurotransmitters, including glutamate, GABA, serotonin, and dopamine. Glutamate, an excitatory neurotransmitter, is crucial for synaptic transmission and plasticity in the hippocampus. GABA, on the other hand, is an inhibitory neurotransmitter that helps regulate neuronal excitability, preventing excitotoxicity. Serotonin and dopamine have modulatory roles, influencing neurogenesis, mood, and motivation. Studies on the hippocampus have also investigated how this structure is involved in the temporal representation of memories. The hippocampus can encode not only spatial but also temporal information, allowing memories to be organized in a chronological sequence. This is fundamental for the ability to recall events in order and understand cause and effect relationships. Hippocampal plasticity is an intense research area, with implications for understanding how the brain adapts to learning and experience. Processes such as LTP and LTD are fundamental mechanisms through which synapses in the hippocampus strengthen or weaken in response to activity. These processes depend on changes in gene expression, protein synthesis, and cytoskeletal reorganization. Research on the role of the hippocampus in depression has revealed that chronic stress can lead to structural and functional changes in this region. Reductions in hippocampal volume have been observed in individuals with depression, and these findings are associated with deficits in memory and learning ability. Research suggests that interventions promoting neurogenesis and synaptic plasticity in the hippocampus may be effective in treating depression. The hippocampus also plays a role in imagination and simulation of future events. Studies show that individuals with hippocampal damage have difficulty imagining detailed future scenarios, highlighting the importance of this structure in the mental construction of possible events. This ability to simulate the future is crucial for decision-making and planning actions. The hippocampus is involved in sleep regulation, especially REM sleep, which is associated with memory consolidation. During sleep, the hippocampus interacts with the cerebral cortex to reactivate and reorganize memories, strengthening synaptic connections that support information retention. Sleep disturbances can negatively affect hippocampal function, and consequently, memory and learning. Research has also explored how the hippocampus is affected by different stages of human development. During childhood and adolescence, the hippocampus undergoes significant changes that are critical for cognitive and emotional development. In old age, the hippocampus may experience functional decline, but synaptic plasticity and neurogenesis can be maintained with cognitive and physical activities. In addition to its importance in memory and spatial navigation, the hippocampus has been implicated in broader functions of contextual information processing. It helps situate new information in an appropriate context, facilitating understanding and interpretation. This is fundamental for associative learning, where different stimuli are linked to form a cohesive memory. Behavioral and neuropsychological studies have investigated how the hippocampus contributes to different types of memory, including episodic memory, semantic memory, and spatial memory. Episodic memory involves recalling specific events and their contextual circumstances, while semantic memory involves factual knowledge. Spatial memory refers to the ability to navigate and remember locations in the environment. The role of the hippocampus in semantic memory is less direct, but still significant. While episodic memory heavily depends on the hippocampus for initial formation, semantic memory appears to be more distributed throughout the brain, with the hippocampus facilitating initial integration of information before it is stored in other cortical areas. Research on the hippocampus also has relevance for understanding the mechanisms underlying anxiety disorders. Studies suggest that the hippocampus is involved in regulating fear and anxiety, especially in contexts that require risk assessment and decision-making. The interaction between the hippocampus and the amygdala is crucial for these functions, allowing the brain to respond appropriately to threatening stimuli. Genetic research has identified several genes that influence hippocampal development and function. Polymorphisms in genes related to neuroplasticity, such as BDNF, brain-derived neurotrophic factor, have been associated with individual variations in memory and vulnerability to psychiatric disorders. These findings highlight the importance of genetic factors in determining hippocampal health and function. Moreover, the hippocampus has been a focus of study in aging and neurodegenerative diseases. Alzheimer's disease in particular is characterized by the accumulation of beta-amyloid plaques and tau tangles in the hippocampus, leading to the deterioration of its function. Understanding the mechanisms underlying these pathological changes is crucial for developing effective treatments. Cognitive and behavioral interventions have also been explored as ways to maintain hippocampal health. Memory training programs, regular physical activity, and engaging in mentally stimulating activities have been shown to support hippocampal function and promote cognitive resilience, even in the presence of age-related changes. In summary, the hippocampus is a vital structure in the limbic system, essential for memory formation, spatial navigation, and emotional regulation. Its ability to adapt and reorganize throughout life, influenced by genetic and environmental factors, underscores its significance in maintaining cognitive and emotional health. Understanding its complex functions and interactions with other brain regions continues to be a major focus in neuroscience, with implications for treating various neurological and psychiatric conditions.